Good evening. I'd like to thank you very much for coming to tonight's Midsummer Night's Science Lecture. This is our last of 4 lectures this summer. My name is Megan Rokop, and I'm the Director of the Educational Outreach Program here at the Broad Institute. And it gives me great pleasure to introduce our speaker tonight, Dr. Deb Hung. Deb is both a chemical biologist and an infectious disease physician. She received her bachelor's degree and her PhD from Harvard University and her MD from Harvard Medical School. She is a core faculty member at the Broad Institute, and she is also an assistant professor both at MGH and at Harvard Medical School. Tonight, Deb will be speaking to us about the critical public health issue of bacteria developing antibiotic resistance and how new genomic technologies may enable us to find the discovery of new antimicrobial therapies. So please join me in welcoming Deb Hung.
Thank you. So I thought that I would try to make this talk as authentic as possible. So I thought I should come down on with my own infectious disease. So the good news is that I've done this before without a voice, so I think I can manage. The bad news for you guys is it's a much smaller room, and you guys are sitting much closer to me.
How contagious are you?
So what I want to talk to you a little about today is really this problem of, number one, infectious diseases, and the problem of resistance, and why, in a lot of people's minds, a lot of physicians' minds, we're losing the war against bugs. So let's just start with what exactly the problem is. So this gives you a sense. There are about 56 million deaths per year in the world, worldwide. But if you look at the breakdown and distribution of the causes of death, you'll see that infectious diseases is here, just 27%. It's the second major killer, only recently surpassed by cardiovascular disease. And in fact, some of these cardiovascular diseases, some of these respiratory diseases, actually are due to infection, and if you group them in there, infectious diseases is still number 1. But infectious diseases also highlights another aspect, and that is really the inequality in the world. Because if you look at the breakdown in causes of death in developing nations, you can see then that infectious diseases becomes much more prominent here at 45%. And so really, combining all of the data, it suggests that almost 1/3 of total deaths worldwide is due to infection. So let's take a look then. What is the breakdown? What types of infections are people dying from? So here you can see that the majority of people are dying actually from acute respiratory infections, hopefully not like mine. But really, very standard things like influenza, pneumonias. Next here is AIDS. The third here is, again, diarrheal diseases. Seems really kind of very run-of-the-mill, very common, and yet it's one of the major killers worldwide. Here's TB. This counts for about 2 million deaths per year. Malaria, and then measles. And what's really terribly heartbreaking about these kinds of statistics is that, quite frankly, the majority are curable, if not treatable. In the majority of bacterial pneumonias we can treat. AIDS, we certainly can't cure, but we can certainly give antiretroviral therapy and manage it. Diarrheal diseases, TB, all of the malaria, we have antibiotics. But let's kind of take the heartbreak a step further and think about how infectious diseases targets children. And in this regard, this really has a lot of significance for the future of the world. So if you look at the percent of deaths in children in each of these areas, you can see that 77% of children in Africa die from infection. It's much less in the United States and Europe, but really all the areas where there's areas that are undeveloped, where there aren't simple things like water sanitation, the rates are much higher. And let's just kind of translate this into very concrete terms. When we hear, a child who dies here in Boston in the United States, or we hear, a family who's lost a child, all of us kind of shudder for a moment. But if you look at the statistics, you get a sense that in areas like Kenya and Malawi, in Africa, this suggests that 49 to 52% of women in childbearing age have lost a child. That means that almost 1 in every 2 mothers in these areas have lost a child due to infection. But it's not simply the toll on lives, but infections also have an incredible economic burden to the world. So this really gives you, again, a cross-section of just what happens in 1 year. But due to malaria, there's $500 million in direct costs to sub-Saharan Africa annually. Here, AIDS is $14 billion per year spent in prevention and healthcare worldwide annually. And this data is old. I mean, this is 1997, 1999. You can imagine that this number is much higher. You know, even things like cholera, which seems incredibly trivial, $770 million lost in 1 year in 1991 in Peru. On the other hand, things like smallpox vaccine and eradication have resulted in $1.07 billion in savings. Similarly, polio vaccine, and I was getting close to eradication, but unfortunately we missed it, also has resulted in savings. So what can we do about infection? And it really is quite a bit, because you can compare the death in terms of infection somewhere in the United States versus what, somewhere in sub-Saharan Africa. You realize there actually are a lot of interventions, and there's a lot of ways that we could prevent these. For example, childhood vaccinations are very effective. Bed nets. In fact, movements like the Gates Foundation, which is really pushing to make sure that bed nets are used in a regular manner in different countries and different areas, have made a significant impact on the morbidity and mortality of malaria. There's other measures such as DOTS, which is directly observed treatment, and that is that if someone comes down with tuberculosis, if we make sure that we observe them taking the medicine every day so that the medicines actually work, that the person can actually be cured, you actually can save up to 60% of tuberculosis deaths. Other types of kind of integrated management of childhood illness means things like nutrition, teaching people hygiene, sterilizing water, antibiotics, which we'll get to, and then HIV prevention strategies. So what I'm going to talk about the rest of the time is really this concept of antibiotics, what a difference it makes, and why we might be headed for trouble due to some recent trends. So, I'm sure we've all heard of the infamous discovery of penicillin by Sir Alexander Fleming in 1928. Actually, he gets all the credit, he gets all, the big name and all the attention. But in fact, the idea of using molds that potentially produce antibiotics and basically secrete them around actually goes back much, much longer than that. And there's reports, from Egypt, from Greece, where people have known that if you take mold and you spread it on a wound, people actually get better. In fact, people had actually recognized that certain molds made, in fact, this antibiotic, but Alexander Fleming was actually the one who found the compound, found the small molecule chemical that was subsequently named penicillin. And resulting from that, antibiotics have saved millions of lives and eased the suffering of patients of all ages for more than 60 years. These wonder drugs deserve much of the credit for the dramatic increase in life expectancy in the United States and around the world in the 20th century. And because of the wonderful success of antibiotics, the U.S. Surgeon General in 1967 said, look, it's time to close the book on infectious disease. We think we've got it conquered. Look, we've got antibiotics. So, how did we get from that statement of it's the time to close the book on infectious diseases to this period now in 2008, where the Infectious Disease Society of America says that we are in crisis? How did we get where the World Health Organization says that we're in a state of emergency? And it's really because we're standing at a time when we have this, very unfortunate convergence of emerging and re-emerging diseases. With technology, what we're able to do is we're actually beginning to be able to detect and diagnose people when people are dying from infections we never heard of. What does that allow us to do? Well, we discover new and emerging diseases, and so we're always playing catch-up. The second is this increasing rate of resistance that we're seeing to almost all the antibiotics we have. And then the last one, which is really an unfortunate one, that stemmed after 2001, which made us all much aware of the fact that that people can use infectious agents as weapons, and they can weaponize them. Even before 2001, there's many documented cases in which people have used bacteria as weapons, but it certainly came much more to the front after 2001. So let's just start at this concept of why we're always losing, and fortunately why infectious disease doctors will always be employed. Is because there's, number one, there's always new pathogens that are rising. So this is from 1977 to 2004. This is a list of new pathogens that that we've discovered that cause disease. Looking around the room, I think that many of us actually will recall when we started reading the newspapers about such phenomena. And for those of you who were around 1977, but basically this Legionnaire's disease surfaced out of Philadelphia where there was a Legionnaire's convention, and many of them came down with this lethal pneumonia that was actually transmitted through the air conditioning ducts of where these folks were staying, and that resulted in identification of this bacteria, Legionella pneumophila. I don't know if you remember the toxic shock phenomenon that happened around the 1980s that was associated with certain tampon use. But you can go down this list, and a lot of them are going to sound familiar. Borrelia burgdorferi, Lyme disease, a little close enough to Cape Cod and Martha's Vineyard to know about that. What else is on here? Bird flu. Again, it's something that we hear a lot about in the newspapers, increasing incidence. So we're vigilantly watching to see, is it going to start? We know it can jump from birds to humans, but we're all waiting for, and with very great apprehension, is that it somehow should mutate so that it could jump from human to human. Then, and that kind of transmission is allowed within the population. Then the ability to control that is going to be much more challenging. And then finally, as we all remember, in 2003, SARS came out. So again, this is just another way to look at kind of emerging and re-emerging infectious diseases that had occurred between 1994 and 1999. You see, nowhere are we immune. I mean, there's also old infections that we thought we could handle, like cholera, diphtheria, even E. coli. But again, they're still causing epidemics. So what's the second problem that in combination with the fact that there's emerging infectious diseases that really gets us in trouble? And it's this problem of antibiotic resistance. So this gives you a sense. It's basically a timeline. Above the timeline is when the antibiotic was deployed. That is when it was first used clinically. Below the timeline is when resistance was demonstrated. And what you can see is that for every antibiotic that's above the line, there's the matched pair below the line. In other words, there is no antibiotic that we have not already seen resistance to. And so just kind of to pull out a few examples to give you a sense of what this means, here's malaria in Thailand. So the malaria drug that we actually used to use all the time it is called chloroquine and sulfadoxine pyrimethamine, two different ones. And in Thailand, all malaria is resistant. So for those who are traveling there, they cannot get these drugs that we very commonly use. And what you do see then is you see 45% that are resistant to second and third line drugs. Here's TB in Portugal. And again, what you see here is at least 13 plus 4, so 17% of TB in Portugal shows resistance to some TB drugs. And now, recently, we hear reports of XDR TB. So we remember, I think it must have been about a year ago, hearing about this lawyer from Atlanta, Andrew Speaker. People remember that, hearing that in the news? So the frightening part was that he was diagnosed with extensively drug-resistant TB, which means that we treat TB with 4 drugs. It's resistant to 2 of the major ones, plus 2 of the minor ones and 1 of the IV ones. When there was one of the first outbreaks of XDR TB occurred in South Africa. Of 53 people who were diagnosed with it, 52 died. When you get XDR TB, the mortality rates are up towards about 80%. So you can imagine what we've gone from is taking an infection which we can treat, albeit it takes a long time, to being utterly helpless. Here's another example of Staphylococcus in Japan. Over 60% are drug resistant, but we'll be talking more about that a little bit later. But we've talked about malaria, TB. There isn't all that much in the U.S. But you know what? The U.S. is not immune to this problem of resistance. This basically gives you a graph of the percent of incidence of resistant strain that have occurred from 1980 up to about 2005. So here, have people heard about Staph aureus or MRSA in the news? So again, Staphylococcus aureus is this very virulent bug that can infect skin, tissues, lungs, hearts, really everywhere. And to first-line drugs, here's the incidence of resistance in 1980. Now we're pretty close to 60% resistant. Here's two other ones, two other types of bacteria. Again, all are on the rise. And what we thought was fortunate was that most of this resistance was limited to hospitals. Right? In hospitals, you go in, you get treated with antibiotics, and other sick people, so that gets spread around. What we learned from that is that about 2 million people per year acquire bacterial infection in a hospital. That means that you go there and you pick up this infection at least 3 or 4 days into your hospital stay. 90,000 people die from these infections that they acquire in the hospital, and 70% of these infections are from bacteria that are resistant to at least one antibiotic. The cost of this is over $5 billion per year in the U.S. Why? Well, because when we go to second and third line drugs, they're more expensive, a lot of them are IV, and so it requires a lot of infrastructure to maintain it, and oftentimes there's longer hospital stays. So what's a typical story about how resistance occurs? So let's take Staphylococcus aureus, this bug that we've heard a lot about in the newspapers, and it's one of the ones that's really highlighted when people talk about resistance. So what happened was in the 1940s, that was when penicillin was given clinically. So even though Alexander Fleming discovered it in 1928, it took that long for people to figure out how to isolate enough of it to actually give it to patients. And so in the 1940s, penicillin kept the bugs under control. It worked great. All of a sudden, when people got a cut or a wound, particularly in the war, it wasn't well, let's just see and hope for the best. Whether this person will live, and we knew we could treat it. In 1942, though, the first Staph aureus bacteria was identified that was penicillin resistant. By the late 1960s, greater than 80% of this bacteria was resistant. Fortunately, in 1961, another antibiotic was introduced, which is called methicillin. So that kind of helped save us during this period when this 80% number was creeping up. But by 1974, 2% of this bacteria is now resistant to the new drug, methicillin. By 2002, this has jumped to 57.1%. Luckily, again, we'll backtrack, and in 1958, vancomycin, another antibiotic, was discovered and introduced that would treat this staph that was resistant now to penicillin and methicillin. In the late 1990s, we begin to identify bugs that are partially resistant to vancomycin. By 2002, we can report 2 cases that are fully resistant to vancomycin. And you can see where this trend is going to go based on what's happened here with penicillin and methicillin. The last frightening thing that's happened with this methicillin-resistant Staph aureus is now it's appearing in the community, not associated with people in hospitals. And so now, the resistance problem is not limited simply to those who are kind of frequently in and out of hospitals, those who are sick or debilitated, but it's happening to young, healthy people. And a lot of this community-acquired Staph aureus was actually first recognized in healthy young college students, who were getting it from the gyms that they were going to. A lot of them, there was an outbreak among college residents. And here, there was one report out of the University of Texas that they cultured 7 children and found 70% of them carry this resistant bacteria. So the question is, then can we always count on the next bacteria? We went from penicillin to methicillin to vancomycin, right? Are we still okay? Are there ones coming down the pipeline? Well, the IDSA, the Infectious Disease Society of America, recently made this statement, which is that infectious disease physicians are alarmed by the prospect that effective antibiotics may not be available to treat seriously ill patients in the near future. There simply aren't enough new drugs in the pharmaceutical pipeline to keep pace with drug-resistant bacterial infections, so-called superbugs. So let me tell you, from my own experience, So in addition to being an infectious disease doctor, I actually am also a doctor in intensive care units. So I talked to a lot of family members about, the patient, and I've had a lot of conversations about, I'm sorry, there's nothing left we can do. A lot of times, I say, I'm sorry, there's nothing left we can do because someone's heart is failing, someone's kidneys are failing, someone's lungs are failing. But I have to tell you that the times I've had to tell patients' families that I'm sorry, your loved one is going to die because we've run out of antibiotics is the one where I find people are most stunned. And even then, take it a step further. If someone's heart fails, at an extreme, we have mechanical hearts, we give people heart transplants. People's kidneys fail, we can give them a kidney transplant. When we run out of antibiotics, when they get that resistant bug, there's no greater feeling of helplessness. So this is a real problem. So can we always count on the next antibiotic? No, I don't think so. In the last 40 years, if you remember that timeline that I showed you, I don't know if you noticed basically the pattern, but I'll point out that in the last 40 years, we've only had 2 new classes of antibiotics that have made it to the market. Everything else has been a derivative of some other currently used antibiotic, which often mentions cross-resistance to some other antibiotic. Between 1998 and 2008, so a whole decade, only 10 new antibiotics were FDA approved. Again, these 2 fell in it, but again, 8 of these are just derivatives of previous ones they knew. So in 2002, among 80 new medicines that emerged on the market, not a single one was an antibiotic. And in 2004, of 506 drugs in the pipeline, only 5 are antibiotics. In comparison, there's 67 in cancer. So what's the problem? Why are most pharmaceutical companies getting out of the antibiotic field? Quite frankly, it's just not money-making. There's very little financial incentive for them to do so. Imagine that, it's great giving, if you're a drug company, to give someone an antihypertensive, because they usually have to take it for life, right? I mean, why all the interest in Alzheimer's drug, obesity drugs? Well, because there's a great market for them, and often people have to take them lifelong. What's the problem with antibiotics? Maybe they're too effective. You take 3 days, 5 days, 10 days, 14 days, and you're done. So the amount of money that the pharmaceutical companies can get out of that is just simply not that great. Further, once you make a drug, you know that resistance is eventually going to come down to what? 1 year, 2 years, 5 years, 10 years. It's going to be there, and your drug is no longer going to be useful, and it'll come off the market. It's also very technically difficult to find, and we'll talk a little bit about that. And their goal is a very broad spectrum antibiotic, because again, if it's broad spectrum, it kills almost every bug out there, there's going to be more in the market. But bacteria can be very different, and it can be very hard to find one drug that's going to kill them all. This is another issue, and that is because infectious disease doctors, such as myself, we know that there's limited lifespan to any new antibiotic because we're going to see resistance. When a new drug is out there that works, we try to save it. We try to save it for only those cases that we know are really going to need it. And so there is a huge market out there, which is why a lot of times you see advertisements now for new drugs, and that is the drug companies are kind of bypassing the doctors and trying to go straight to the public, and getting the patient to call up their doctor and ask about the purple pill. The other issue is that the median time from when you find a drug, much less get it tested, show it's going to work, and get the FDA approval, is 6 years for testing and FDA approval, almost 8 years even to get to clinical testing. So what we're saying is, you're talking 14 or 15 years. Is it really worth it, given all of these limitations? And you know that only about 1 in 5,000 drugs that they do clinical testing are actually FDA approved. So now so that just, I hope, gives you the framework of the issue. Is that really, look, infectious diseases are still a major killer in the world. There's a problem of kind of continuously emerging new bugs, but there's also this huge problem of resistance. And we're simply not making enough antibiotics to counter it. So what are we trying to do? What can we do? We're going to switch over then and talk a little bit about the science of say, antibiotics. So we're going to start with this very simple standpoint, and just so we're all on the same page, and that is the biology, right? So we all know that there's a cell, for bacteria, a human cell, and the dogma, right, is everything's encoded in the DNA, in the genome, that's converted to mRNA to protein, right? Everybody's familiar with that? So it looks something like this. So here's DNA, and it's all coiled, right, in the human cell, and here's what it's stretched out. It's this double helix. Can you see this? Are there lights? Can you turn off?
We can see it.
We can
Okay.
see it, don't worry.
So what's happening here is that, enzymes or proteins, which are the workhorses, basically replicate the DNA, and here it's making mRNA, all right? And the mRNA piece breaks off. It's just adding a tail. So here's the DNA, here's the mRNA, comes out of the nucleus into the cytosol. And then what happens, right? From mRNA, we make protein. So again, these are proteins, it's a ribosome, but it's a whole synthetic factory that makes these long chains of amino acids that are encoded from the mRNA. And you get these proteins that fold. So these are all what's essential for a cell, right? You have to be able to replicate your DNA, right, so that you can divide and form new cells. You have to be able to convert your DNA to mRNA. And then from RNA, you have to make protein. And then proteins, right, such as these, really the workhorses of the cell. They're the structural components. I mean, it's like saying kind of they're the beams and walls of the building. But they're also the worker bees. I mean, they're the enzymes. They do all the work around here. They do what Megan does. They do what I do in this building. We run around and do the work. And so you can imagine then that it makes a lot of sense that antibiotics target these functions which are essential in bacteria in order to kill them. So what are the mechanisms of the current antibiotics exist? Well, there's antibiotics that inhibit DNA or RNA synthesis. A common one that you might have heard about is Cipro. Again, that got a lot of attention around 2001 with the anthrax scare because everybody was asking about for Cipro. Another way is to inhibit protein synthesis, and that is preventing the bug from going from RNA to making the structure and enzymes that are required for the cell to live. You can inhibit cell wall synthesis, right? I mean, there's a wall that encloses the bacteria. They can't make the wall. They can't survive. And then there's some other new, this is an old one, which inhibits making the nucleotides that are incorporated into making the DNA. And then there's a new one that somehow affects the integrity of the membrane between cells, of which we don't know. So I have to say that when I look at this is the striking thing. All the antibiotics we have only target 5 steps in survival of a cell. We now know, based on genome sequencing and genome analysis, that let's say a bacteria has about 4,000 genes. We know that about 300 or 400 of those genes is required for the cell simply to live. I mean, we're not talking about doing anything fancy like causing an infection. Simply to live in a test tube requires 300 to 400 genes. And yet, we've only managed to target very few. Why is that? We don't know. I will say that almost all the current antibiotics we use are actually isolated from nature, the same way penicillin is. So bugs out in nature have secreted and made these antibiotics, which we then identified, right, and purified or synthesized, and we give them as antibiotics. So is this nature's way of telling us that these are the main ways to intervene and kill a cell? And it's simply through evolution and natural selection that those bugs which have survived, which make things that inhibit these and kill their competitors, are the ones that have survived. So that's how antibiotics work. How does resistance occur? Well, there's 2 major ways. One is the idea of mutation. So remember all the DNA that was kind of wrapped up in the genome, right? And it goes from DNA. DNA encodes RNA, which encodes the protein. So what can happen is, if you have a mutation, if you change one of the bases in the DNA, that will change one of the bases in the RNA, which will then change one of the amino acids in the protein. And instead of getting a protein that's a nice Pac-Man, you might get a protein that has a bump on it. And then this triangle where the drug no longer fits into where it should go. And if it doesn't fit, and the enzyme can still work, you can see this drug no longer works, the cell will live. The second major way that we get resistance is what's called horizontal gene transfer. And that is that instead of dealing with kind of the whole genome of DNA, bacteria have figure out how to keep small plasmids or small pieces of circular DNA, which they can then basically just pass from bug to bug. And what that means is very often that if a bug picks up a resistance, a piece of DNA that has resistance to many different markers, it can basically then just hop to the next one. And what these pieces of DNA on the plasmid often encode are, one, either efflux pumps, which is basically, it encodes a protein that now inserts into the membrane and basically pumps the antibiotic out so the bacteria doesn't even see it. It can make proteins that will basically degrade the antibiotic, destroying it, or it can modify it, change the small molecule some way, so again, it's not. So, there we are. That's how antibiotics work. This is how resistance occurs. What are we doing about it? How do we find new antibiotics? So let me tell you kind of the standard approach in pharmaceutical companies. And I don't have to go too long. For those who came, they might have heard Robert Gould give a talk a couple of weeks ago about high throughput screening. But the idea is this, can we screen hundreds of thousands and millions of compounds at a time to figure out which ones will actually kill the bacteria? And so the way it's done is we do it in plates like this. This one has 384 little wells where we introduce the same bacteria, and then we can introduce a different chemical into each well. And then we can basically, have some type of readout and look and say, oh, well, look in this well, the bugs died, and we know what compound we put in there. There's other formats people can now put in 1,536. People are going to even higher and higher numbers, but this is the way. And then you use robots to basically, add the cells and bugs to the plate, and then you add compounds. What kind of compounds can we screen? Well, there's 3 different kinds. These are drugs that kind of are commercially available. These are very common drugs that treat humans. There's also these natural products that I told you about that are made by bugs out in the environment. And then there's molecules that people are trying to synthesize completely artificially. So what kind of assays do we use in those little wells? Well, there's either target-based or there's the whole cell screen. The whole cell is kind of what I already described to you. What you do is you take the bug, and that has all its different proteins, you put it in the well, you introduce a different molecule in that well, and then look for molecules that inhibit that essential protein, and then the bug dies. The other way is to do what's called target-based, and that is, instead of taking the whole cell, you simply take the enzyme, or you take the protein you know is essential, you put the purified protein in there, and look for small molecules that will directly bind that target. So the catch, though, is if you decide to do the whole cell way, there's a catch, and that is that you can kill the bugs, but you have no idea how the compound is working. Right? You have no idea about the mechanism, and why should you care? Well, because the classic way that we go from finding an initial lead in that well and screening of 1 million compounds to actually making the final drug is that you often try to crystallize the protein with the compound. And this is what that is. It's simply a crystal of purified protein with a small molecule bound. What we do is we try to solve the structure and get a sense of what this looks like. And so you can see, again, these curves is basically the chain of the amino acids that are all folded up now in a protein. And I don't know if you can see this, but there's a small molecule here that's basically docked. And it's docked into the protein. And once we get that picture, we can say, oh, look there's a little bit more space up here. We can add a little bit more to that molecule and get it to work even better. And so we can make changes to our initial molecules in rational ways based on what it looks like in order to make a better drug. But going back here, if you find the drug simply from screening whole cells, how in the world do you know how it works? And so this is where genomics comes to bear. And that is, genomics allows us to find the needle in the haystack. Remember, 4,000 genes in bacteria, 4,000 proteins. There's potentially 4,000 different proteins that this drug is binding to. How do you find it? What we do is we take it, we generate resistance, right? Remember that you can get a mutation that then prevents the bacteria, the more protein from binding. So how do we find out which protein carries that mutation? We go back and we sequence the DNA of the mutant bug, right? So we can see that there was a change, for example, from AGCCTP to AGCCTG, and you see this difference between the parent sensitive one and the mutant one. And so you know, in fact, this gene is a target. Once you find the gene, you can go back, identify the protein, crystallize it, get the structure, and then you move on. And so does this work the way that industry has been doing it? Well, based on the pipeline, and I told you that only 2 new compounds have appeared on the market, 2 new classes in the last 40 years, you can see it doesn't work that well. So this is an experience of GlaxoSmithKline, which is one of the big pharmaceutical companies, from 1995 to 2001. So they performed 70 of these chemical high-throughput screens. Each time, it cost them about $1 million per screen. They screened about 260 to 300 or 530,000 compounds in each screen. They targeted 300 genes in this type of approach. They did 3 whole cell screens. And at the end of this, so at the end of about $60 million, no real viable candidates. So can you understand why pharmaceutical companies want to get out of the antibiotic field? So what I want to suggest to you is we have to do something, right? Resistance is rising, new bugs are coming, and we've got virtually nothing in the pipeline. So what I'm going to try to show you in the next couple examples are potentially ways to start thinking outside of the box and potentially new paradigms of what you might think an antibiotic is or how we might discover a new antibiotic. So remember, all up until now, right, all current antibiotics, the way that people have been doing it is they're simply looking for compounds that will kill the bug, right? It'll kill, we can have the bug sitting in a test tube, it'll kill the bug. But it's not really what we care about. What we care about is finding a compound that will kill the bug in a test tube. Not really, right? What we care about is treating the bug here in the patient with cholera, here in the patient with TB, here with the patient who's in the hospital and has gotten one of these bad resistant infections. So if that's what we're interested in, then you realize that what we should be thinking about and the biology we should be understanding is not simply what is essential for a bug to survive in a test tube, but what we really want to know is what is this complex interaction that goes on between the bug and the human host. We know that somehow the human host can have an immune response that can clear the bugs, right? Lots of people can get some type of infection, or they get antibiotics and it clears. Somehow we know that the person recognizes the fact that there's a bug in them so that you can stimulate an immune response. Somehow we know the bug is doing something to the host to cause this tissue damage. And this is the complex relationship that we're trying to understand, because what we're trying to do is disrupt this interaction, not simply kill bugs in a test tube. And so what is the complex dance that goes on between the bug and a human host? Well, the bug, quite frankly, has to encounter the host, it has to enter, it has to be able to spread and multiply, it has to cause damage, right? And then it has to be able to be transmitted to the next person. What does the host have to do? Well, it has to be able to recognize the infection, it has to be able to clear it. It's not just some random thing that occurs, it's actually very carefully orchestrated. So this gives you a sense, these are red blood cells, and this is a white blood cell. This is an immune cell in humans, and it recognizes a bacteria. And it's not just some random clearing, it actually chases it down. You can imagine, right? This is an awfully complex process. A lot of biology is going on here. Eventually it engulfs it and clears it. So you can see, right? Everything that the bug does, everything that the host does, is all extremely carefully orchestrated. And so the question is, can we disrupt this tight regulation, right? Are there alternative ways that we can prevent infection or treat infection other than simply killing the bug in the test tube? And this is what I'm trying to argue, right? There's a balance between the host and the bug. We're not saying that, well, one way we've tried is simply kill the bug. Sure, that would be easy, but finding new ones is very hard. Are there other ways that we can basically tip this balance, right, in favor of the host? And so 4 potential new paradigms is this concept of antivirulence drugs, targeting in vivo essentials, that is, functions of the bug that are essential in the host, which you'll never see because they don't require that in a host test tube. Can we modulate the host and its immune response? And can we develop compounds or drugs that might prevent the development of resistance to an antibiotic? So again, this is another way to think about that complex interaction that goes on between the bug and the host. right? You saw the bug runs, right? So most bugs, in order to cause infection, have to be modal. They have to get to where they need to be in order to cause infection. You know, if you drink cholera, the bugs have to be able to swim through all that stuff in your intestine, has to be able to swim through a mucus layer right next to your intestinal wall and get through to the intestinal cells right lining your intestine. So as they get there, it has to adhere, has to actually stick to those cells, and then it has to know, you know what, I'm in the right place. I can do something bad now. I should make my toxins, and I can cause disease. So you have to be able to make toxin, you have to deliver it, and it has to have its function. We could potentially interrupt all of these. So let's talk about antivirulence then as a way of stopping. So this is that example I told you about, Vibrio cholera, right? Lives out here in the environment, here in Bangladesh. Somehow it gets ingested into the human host. That's to pass through the stomach, get to the intestinal epithelium, multiply, then it makes its toxins, right, which is what gives people the horrible diarrhea that they have with cholera. And then the bugs are now in the diarrhea, they secrete out into the environment so they can infect somebody else. So the question is, most antibiotics, what would they do? They try to kill the bugs, okay? We can do that. If you give it, that will happen. You'll get resistance, that will happen. We've seen that. Step out of that mode for a minute and think, well, look at this whole complex interaction. Can't we stop it in any other way? So what we're trying to do is say, you know what, let's not worry about killing the bug. Let's simply try to disarm the bug. If we disarm the bug, can the host immune system do the rest to clear it? And so what we did was, we found compounds that prevented the bugs from making toxin. So you have bugs, they just don't make the toxin that they need to cause disease. And what did we do? We fed it to mice who we had infected with cholera. So we take the cholera bugs and the compound and the drug, and we feed it, we orally gastrically inoculate it into mice. And the way we tell if the compound, if anything is working, is after 18 hours, we collect the small intestine, we grind it up, and we plate it out, and count how many bacteria are there. If you do that, and this is basically, it gives you a sense, it's actually the log of number of bugs you can recover. If you take a strain, I'm not going to go through all the details, but you take a strain, you feed it into mice, but don't give them a drug, you recover 10 to the 6 bugs. If you give them a drug, except for 2, it's completely gone. So you can see that this type of strategy actually works, right? Simply disarming them and preventing them from making toxins or making their weapons, which they require to cause disease, the immune system will actually clear the bugs. What about in vivo essentials? What do I mean by that is, look, in order to perform all of this, there's hundreds of genes that are required. All right? Remember, everything is so carefully orchestrated. So how do we target any of these? Well, how do we find compounds that do that? Ideally, right, you would take your million compounds, have an infected individual, start feeding them one at a time, and say, oh, well, look, this one worked. And you found it. Obviously, that's not feasible, nor would any FDA panel approve such an experiment. Right? What about a mouse? Can you do that? Well, let's infect mice and just give them a different compound until we've gone through a million. Again, it's kind of expensive to grow up a million mice. Remember, we do this in plates, in small plates like this, which are about the size of a mouse. How do we get it so that we can actually screen in one of those tiny wells in these plates? Well, we need something. If not a human, if not a mouse, we need some host that's a model that we can use and infect. And so there's 2 different model systems that we use. One is C. elegans, which is a nematode. It's a worm. And you know what? 10 of those fit great into one well. The other is this Daniel-Ririo. It's a zebrafish. And the zebrafish embryos, here you can see this is 200 microns. So again, several of these fit very nicely into a little well. So what you do is you can infect these guys, right? Now introduce compounds in the well, and then try to pick out which well you actually could save these guys from infection. And this is what it looks like. Is we can do it by microscopy. And this is a microscopic image. Basically, the bottom line of this is, you see, these are the worms. When they curl up, it's because they're alive. They're actually moving and swimming. When they're straight rods, they die. So you can imagine, right now, in these wells, we can take, hundreds of thousands of images of different compounds in these different wells, and simply ask a computer to tell us, right, which compound actually saved the world. This is what it looks like when you use zebrafish. It's not curved because they can move, because they're long, or rod when they're dead. But basically, what we do is we infect them with fluorescent bugs. And so you can see that there's nothing here. Well, this is after 4 hours after infected. But here, as the bug and infection spreads, right, we can see the fluorescence in the fish of the bug. And then here, you can see it die. So again, you can do this type of screen because you can quickly pick up, whether it's managed to save the zebrafish embryo or not. The question is, how good is this? I mean, does this immune system look very much like our human one? In fact, it does. There's the majority of exactly the same properties that human immunity does. All right, so that's how we look for in vivo essentials, right, through these different animal models. A word about the host. So can we modulate the host? Let me talk about TB for a minute. A third of the world's population is infected with TB. Right? Many foreigners who come to our country, right, when they get, I don't know if you guys have all had that PPD tested, right? They inject a little bit and see if you get one. Right? Those people with lungs, they're infected, a third of the world. But only 2 million people die of TB each year. Why is that? It's because the mass majority, over 90% of people, can get infected, but they never get disease. Which means the immune system is actually pretty good. Is there some way we can exploit that? Is there some way that we can boost right, human immunity and have that help clear the infection? And so, here's an example. Remember that cell that was chasing the little bacteria? Right, that's an immune cell. This is an immune cell. I'm just blowing it up. This is a macrophage. This is the main line of defense in us when we get infected with TB. Most macrophages, what it will do is, here's the bacteria, right? You remember the video where it engulfed the bacteria? When it engulfs it, it actually is able to then have a process of killing the bacteria in this endosome. Bacteria gets degraded and eliminated. What happens in people with TB who go on to have disease? What happens is it gets engulfed, and you know what? There's something wrong. The macrophage isn't killing the bugs. And in fact, what happens? TB replicates in the bugs. And eventually, when this cell lyses, TB then spreads and goes to the next one. So how do you get this and not this? How do we identify? So it's the same idea. Can we screen in 384 well format and find those compounds that will basically help the macrophage clear the bugs? And so this is what it looks like. Here, the blue is a dye we use, but the blue basically stains the human cells. The green are, again, fluorescent tuberculosis bacteria that we made. And when you infect, basically put the cells into place, and in infect, what happens here, you can see the bugs have replicated and they turn green. But if you could add a drug that would help the macrophages kill the TB, you would see this instead. So the question is, can we screen using a microscope and a computer to tell us compounds that have given us this? And so in fact you can. So again, in 384 well format, you can put in bacteria, you can put in cells, we add a different chemical to each well. We use automated microscopy. Again, Megan, I don't know if you see this. Right, what we do is we have automated microscopy that we can look at hundreds of thousands of images, and we basically teach the computer. And again, Ann Carpenter, my colleague spoke about this last week, we can teach the computer to analyze these hundreds of thousands of images and pick out the one that looks like this, right? Where it's all blue cells, there's no green, there's no bacteria, from the mass majority of where the compound has no effect and TB can replicate. So we can, in fact, identify small molecules that prevent TB replication in human cells. So again, yet another approach. Finally, I'm going to talk a word about anti-resistance approaches. And so these drugs that I'm going to talk about are a little bit different, right? These, you can imagine, it's really truly trying to clear infection and trying to tip the balance towards the host. This last one I'm just going to talk about because it's just a little bit of a different concept of, even if we had new drugs, aren't we just going to get resistance? Is there some way where we can prevent resistance from occurring? So let's review kind of the biology we talked about, and that is resistance. So remember, there's bugs. There can be a few bugs where they carry a mutation, right? So genetic difference in that bug, in the genome or in the DNA, that makes it resistant. So what you do is you add antibiotics, you kill away everything except the resistant ones, and then those grow up again. There's another phenomenon that's actually called drug tolerance or phenotypic resistance. And you don't hear much about that, but it's actually it's the dirty little secret, is that if you have a population of bugs, if you have a flask of bugs, if you treat with antibiotics, even if they're all sensitive on the genome level, a few bugs actually manage to survive. If you take those few bugs and you grow it up again, you'll get all sensitive bugs again. But if you kill again you'll have a few remaining. What are those bugs? Nobody knows. We're trying to understand the physiology of them and why they can survive. Are they relevant? We think so. I mean, think of chronic infections where we treat with antibiotics a long time, right? We can isolate bacteria that are not this, right? They're still sensitive, and yet it's really hard to clear. You might have heard people get urinary tract infections. People can get what are chronic. You get them over and over and over again. You have to treat them over and over and over again. You never clear. And we think that has a lot to do with this population. So if somehow we could figure out how to eliminate this, and both the red cells and the black cells, then we hope we could take the population and clear and get rid of them all. Is that possible? Well, we think that we can address some of it. We think we can address some of it because, in fact, a lot of this resistance doesn't pre-exist here, but what happens is that when you expose a population of bacteria to antibiotics, they actually induce resistance. What do I mean by that? Bacteria are really smart. They've had a long time to evolve to really optimize themselves. And one of the things that they've evolved to do is have what's called an SOS response. If you take bacteria and you stress it, what will it do? If you take one of us and you put us under stress, what do? We try to find our way out, right? And we'll try anything. If there's a fire in here, I'm going to try the door. If I can't get out the door, I'm going to try the window. If I can't get the window, I'm going to try some other way to get out of here. Bacteria do the same thing. If you put them under stress, they try to find ways to escape that stress. If you treat them with an antibiotic, they try to find ways to escape the killing or death from that antibiotic. And what do? They increase their mutation rates. And by increasing the mutation rates, they increase the likelihood that you'll get that change in the DNA, you'll get that change in the RNA, you'll get that change in the protein, so that the antibiotic can no longer bind and won't work. And so this group out at Scripps has actually shown that if you have stress, you activate this whole response system, this SOS response system, that results in activation of a DNA polymerase. The DNA polymerase that basically copies and makes that copy of DNA. But instead of one that's really very good and very careful and puts in the right base, it makes mistakes. And by making mistakes, it generates, the population which can be resistant. And so what they did was they basically have shown, not with a chemical, but with genetics. And so the chemical approach of finding a drug is in the horizon. They've shown with genetics. And if they can infect with something that is unable to activate this SOS system, and now they infect a mouse with an antibiotic, they get no resistance. All right. And so there is potential, if we can prevent the bugs from figuring out they're under stress, maybe we can trick them into being killed. So I'll leave it with this, We all have our different reasons for wanting the host to win. I kind of suggested 4 potential strategies, An antivirulent strategy, targeting in vivo essentials by screening with whole animals, trying to help the host, or trying to find compounds that prevent resistance. Now, I don't want to leave on such a dire note to say, look, there's horrible bugs out there, they don't have enough antibiotics. All I'm saying is, you know what, it requires a really dedicated effort. And we have to be really committed to recognizing the problem and really committed to doing something about it. Because when we find the right antibiotic, they really work well. Here's an example of a young man who has HIV. Given the right drugs, in 6 months, this is it. Okay. Thanks so much for your time.
Prospects of bacteriophage therapy in treating this type of disease.
So that's interesting. So he asked about the concept of using bacteriophage, which is basically, it's a virus. So we always think of viruses as bad, case in point. HIV virus, it's bad. But they're actually good viruses. They're viruses that will kill bacteria specifically. So the question is, can you use viruses to actually kill the bacteria? And the answer is yes. So the Russians have actually, before the dissolving of the Soviet Union, and a lot of the research they did, they have actually studied this a lot, and they had actually identified a lot of viruses that could kill different bacteria. And some work of that is still going on. So for example, people can show that, if you get a soft tissue infection, people have tried injecting the virus around the tissue, and it works to some degree. There's also a story of, must be, I think in the 1940s or 50s, in 2 areas in Southeast Asia, there was a lot of cholera at the time, and there's a lot of Vibrio cholera specific phages. So what they did, what the government did, was they went and they dumped viruses into the wells of where people were drawing water, and they did it in one well, and they didn't do it in another area, so that they could compare. And the problem is that people that they dumped the virus in did so much better than the people where they didn't dump the virus in, that they went and they dumped it in the other one. And so there was never really good controlled study to tell us exactly how well it works. There's a catch though, and the catch is this, that virus is very specific to a certain bacteria. And even if you take, for example, Staph aureus, there's all these different strains out there. And the virus can also be strain specific. So the difficulty is, unless you were to know in real time the exact strain, the exact bacteria, you wouldn't know which phage to give. And that's the difficulty.
Have they tried cocktails and antibiotics?
Yes. To which particular infection?
Well, I don't know. I mean, just has that approach been used a lot of times?
People have. And the problem is that there isn't good data to show it helps in all cases. So for example, HIV, right, there's a case where you give a cocktail of 3 and it prevents resistance from developing. TB, you give a cocktail of 4 and it prevents resistance to a degree. So yes, the answer is giving 2 can be better, but it can also be problematic because now you're showing them 2 antibiotics and they could develop resistance to 2 or 3 at the same time instead of. Yeah.
A lot of the things you talked about new approaches don't really address the central point you made earlier, which is that the drug companies have no incentive to go in any of these directions either. So how do we?
That's what we do.
How do you deal with that?
Well, no, that's true. And so, I mean, I think one is, the NIH and NIAID recognizes this. And so they are trying to find ways to provide incentive to companies to do so. One of the approaches is that a lot of the non-regulated work occurs actually in academics, right? And then you find some way to transition to the industry. NIAID is actually providing funds for collaborations, for academics across industry. And let's hope for the best. I don't know if you were here for Robert Gould's talk, but he leads a team here that is working on novel therapeutics. And so the goal is, again, to take academic finds and find some way to basically go down the non.
Deb, you talked a lot about compounds and stuff. What about the abuse and overabuse of antibiotics?
Yeah, no, that's a huge problem. Oh, I wish I could remember the statistic, but it's something like 70% of courses of antibiotics that are given out are unnecessary. You know, they don't treat what the person has. And that's probably one of the single most important factors for Penicillin resistance. You know, there's, you know, all we can do is educate people not to misuse antibiotics. You know, but it's difficult. You know, in many grade schools, right, or daycare centers, if your kid is sick, right, they can't come. But if they're on antibiotic, they can come, even if they have a virus for which there is no antibiotic. But what happens then is that the parent goes in to see the doctor with the kid, and the chance of walking out with a prescription for antibiotics is much higher if the parent asks than if not. And the parent asks and says, I really need this so my kid can go to daycare. The likelihood of getting that prescription increases high, right? So the education has to really be on every level, to the public as well as to practitioners.
You talked about thinking outside the box and isolating compounds that don't just kill the bug, but keep it from producing toxins. And I get the impression that you've had a lot of success with that, and that was cheaper and faster than traditional methods of looking at new antibiotics. Is that true? Or?
No, I have to say that all these new paradigms that I've talked to you about is, you know, is all in the academic level, and it's, you know, it's all work for the last maybe 2 years. So, you know, when I suggested that it's almost 15 years from start to finish developing a new, these are nowhere near, and these are all only paradigms which we have yet to, you know, establish and show they're very effective and get buy-in from the industry.
So how optimistic should we feel?
I would just say keep your eyes open.
You mentioned, I want to go back to something you mentioned earlier, which is about in Greece and in other countries, you said they used antibiotics to treat wounds, infections. Do you know anything about why they use spider webs to treat wounds in some traditional cultures? Is that also a fungus related? It's not fungus, but is it to have some of the same properties or something?
I
Just thought I asked.
Are all of the mutations bacteria is permanent? Is there any point at which the bacteria would forget
generally no. You know, those of that which have actually mutated their DNA, the simple answer is no, it's fixed it. There's a much more complicated answer, and that is that if that mutation makes it resistant, but it actually cripples the bug a little, okay, then there could be evolution that goes on to repair that in the absence of drug pressure, right? And then it might then be sensitive again. But that would then require maintaining the pressure.
One of the questions that occurred to me as you were going through all of this, in terms of trying to find new compounds in whatever form in all of the different categories, is how do you find things that don't also cause problems for the host? Like,
well, that's a good question. And so that's why from compound to drug takes so long. Because if we screen just against the bug, that doesn't tell us anything about toxicity to the host. Except, right, when we're screening potentially in fish or in worms, we at least get that first pass.
right. But if you're looking for a particular part of the pathway to interrupt a particular process, since bacteria and multi-cell organisms share a lot of the same processes, how do you find something? How do you even come up with a guess of what's going to only affect the bacteria and not the host?
Well, that's why people used to guess, and that's why it didn't work very well, which is why they could go through this, let's just screen 1 million. Right, and then you take those, and the next thing you do, you screen against, mammalian, human cells, those that are toxic you throw out. you just kind of whittle away.
So before we thank Deb one more time, I'd just like to make a couple announcements. The first is thank you again for your patience in changing rooms. The second is we'll still be having a reception down in the main lobby, which is just one flight down. So there's a staircase one flight down, and then there's also 3 elevators that you can take. And then finally, some of you may have gotten flyers at the beginning of this lecture when we're still down in the auditorium. There's a lecture series at Harvard called Science in the News that occurs during the fall semester that's also open to the general public. So I would encourage you all to take a look at the talks that are going to be in that lecture series this fall. And then finally, I'd like you all to join me in thanking Deb once again for being here.